So today is something of a special webinar for me. I have been very excited about this topic, and I know that my team has been as well. This is a collaboration between myself, my science team, and the clinical team, and you will be meeting them at the end, so please don't jump off. When I finish saying, I think it's really important you get to see who the brains are behind 3x4. But launching in, what we're going to be talking about today is science you can trust, and basically how do you get to understand how we do the science at 3x4 Genetics. So let's jump in. This helps if we... Right, so everything in science, nutrition, exercise, sport is an evolution of science. And I've been doing this since the year 2000, and obviously in the 23 years that I've been spending in nutrigenomics, I've seen and experienced a great deal of change. And this is such an old slide, but I couldn't find a new one. But you can see basically from early days how these are really the number of articles that we're seeing in the literature have changed. This is 2017. So you can imagine by 2023 how we're seeing a change in the narrative and the conversation that's happening in the world of nutrigenomics. So that brings us to the concept of methodology. And really, methodology is how do we go about building a genetic test? And in this case, how do we go about building the 3x4 genetic test? And there's a whole lot of elements to it that I'm going to be unpacking today, talking about a lot of the science of what science there is. And then the second part, I'm going to be talking about how do we apply that science in 3x4 genetics. So to jump in, let's start talking about the studies that are available in the scientific literature that myself and my team will be reviewing to be able to figure out how do you go about building a scientific test. So there are a couple that we're going to be looking at today. The first concept I'm going to talk about is the single SNP. We're going to be looking at GWAS and total genotype scores, candidate gene association studies, and then a little surprise at the end, which is my or our favorite kind of study, which we will get to. All right, so I had to start with this because anyone who has heard me speak knows my feelings about MTHFR and what happened really in the first couple of decades of the marketplace of nutrigenomics. So pretty much from about 2005 onwards, we saw this incredible growth in companies that were marketing and selling genetic tests of a single SNP. And I would say the absolute winner, hands down, was MTHFR. And I had another slide I took out, which was a company in Australia for like 70 Australian dollars. You could have the MTHFR SNP done. And as they refer to it is, by the MTHFR mutation, which causes the following diseases. And that was the way they advertised the test. And certainly they're not the only company in the world that have done that. And so what happened over the span of the last couple of decades is we got this understanding that these SNPs like MTHFR and COMT and ApoE, FTO, by themselves were quite extraordinary and by themselves could generate all kinds of recommendations, whether it was a diagnosis for a disease, supplements, interventions, and entire companies have been formulated around MTHFR. They're still very much in existence. And mostly they are supplement companies. And this was really came about as the birth of methylated B vitamins, where we saw MTHFR being associated with very high dose methylated folate. And we all know that doesn't make sense because when you have MTHFR SNP and you take methylated B vitamins, it bypasses methylated folate, bypasses the SNP. So I just want to put in perspective my concern about where we've come from in terms of the single SNP. Now we're going to talk about the concept of polygenic, because if the single SNP is not a solution by itself, then we need to start looking what else is going on in the literature. So let's talk about the concept of polygenic and where it came from and how it exists in the literature. So this article is quite a few years ago where they talked about the prospect of using risk scores in polygenic medicine. And basically, I love this quote, now is the time to start planning for polygenic medicine. So it's not something that I invented. It's not something that 3x4 invented. This is something that has been in the conversation in the scientific literature for as long as there has been genotype studies. And you probably have heard me speak about this study numerous times. It came out in 2020, August 2020, by Farhead. And for me, it was one of those pivotal science papers that really changed something for me about how I reviewed and thought about genetics. And essentially what they did was they said, you know, we've got this concept of high penetrance and low penetrance, as I often teach about. High penetrance being those medical genes that by themselves have a very strong impact on your likelihood of getting a disease. We can put BRCA in there, familial hypercholesterolemia, the FH gene in there. But what they did was they looked at the variance in why someone with a BRCA gene variant would have an 80% chance of getting breast cancer and someone a 40% chance. And we know that just having the BRCA gene, your chances of developing breast cancer is anything between 40 and 80%. So it's like, well, you know, 40% is very different from 80%. So what else is going on there? So we know that part of that is going to be diet and lifestyle choices, but what else is going on? And what I absolutely loved about this research, and this article was actually based on a prior article where they showed that the polygenic environment determined whether or not the monogenic gene would express itself. So what they're essentially saying is having the BRCA gene is one thing, but the polygenic environment, oxidative stress, detox, methylation, anything that would have a kind of a cancer inflammatory susceptibility to it, we're determining whether that BRCA gene would be switched on. Because just having the variant of BRCA, remembering that the BRCA is just a SNP, it's just a SNP. So what would make this gene switch on? Well, they discovered it was this polygenic environment. And I love this because traditionally in genetics, there has been a separation between medical genetics and nutrigenomics, the world that we live in. And this was the first paper that brought it together saying, neither is more important than the other, but actually they speak to each other. And in the paper, you will read that it's not only a BRCA that they looked at, they also looked at Lynch disease, and they also looked at familial hypercholesterolemia, how the polygenic environment of cardiovascular disease will impact whether the FH gene will express itself. So if you haven't read this paper, I highly recommend. Right, GWAS, and I know people call it by different ways. I'm going to call it GWAS, gene-wide association studies. And we started seeing these coming out in the literature in about 2006, 2007. And it was a fundamental shift for the world of genetic research. Up until then, most genetic research was based on twin research, very small sample sizes, very small sets of genes, lots of kind of hereditary and twins, and some candidate studies, which we'll get to. So let's all just make sure that we're on the same page when we understand what is a genome-wide association study. Quite simply, you take a very big microarray, so a big chip with almost like hundreds of thousands of gene variants on, right? And then you look at a disease state. It could be diabetes, it could be hypertension, it could be Alzheimer's, so any disease state. And you have a group of cases and you have a group of controls. And then what you do is you do the genetic test. And what they're trying to see when they do this is on this microarray, in this control group versus the case group, are there certain genes that are showing up, lighting up, right? And that's why you can see in the image. And when you look at the bottom picture, those are all the genes that they're looking at. And you can see on chromosome, I believe it's on chromosome 19, one of the genes in particular is showing up very highly. And I'll show you a real case example of that. So the thing about GWAS is they do not have an hypothesis when they go into the research. They're simply trying to see, are there genes that show up more in a diabetic population than a non-diabetic population? And the kind of negative side is I often call it a fishing expedition because there is no hypothesis or biochemistry or biological plausibility. It is simply, are there any genes? Now, while this isn't kind of helpful at a clinical level, it's extremely helpful when you're starting out in the area, trying to understand what genes might be having an impact. So this is an example of a gene that started out in GWAS, and that's the FTO gene, which I'm sure you're all familiar about. And it stands for, the fat mass and obesity gene. And this was identified many years ago in a GWAS study. And not just one GWAS study, multiple GWAS studies that were using an obese population as the case population, and the control were normal weight, kept on seeing the expression of this gene variant called FTO. And if you look at the image on this one, this is from an actual obesity GWAS, where the red circle is, you can see the gray dots, that is FTO. So of all the GWAS obesity susceptibility loci, what they talk about is just gene variants, FTO had the largest effect when it talks about BMI, waist circumference, so pretty much all of the obesity traits. And they found this against all the obesity traits throughout the life cycle and across diverse ancestries. So this wasn't just Caucasian, this wasn't just African American, this wasn't just Asian. All the different populations found the same impact. Now, I've always had fascination with FTO because I can't get over the name. Fat mass and obesity gene tells us nothing. If you think about MTHFR or ApoE, it gives us a wonderful insight into where in the biochemistry the gene is happening, not so much with FTO. And in fact, for the greatest time, we never had any understanding of what the FTO gene actually did, what the enzyme that it was coding for did, and how it was impacting. We're a little bit more ahead now, understanding the mechanism, but in the beginning, it was included in genetic tests simply because of how powerful it was in the GWAS studies. Okay, so now I'm going to give you, give me a second. I'm going to show you some studies that I have put together that give you some examples of GWAS studies and how well they work, okay? This one is looking at diabetes, where they were trying to understand, can we use a polygenic risk score as a screening tool to be able to understand who is more likely to get diabetes and not? And in this, it's actually a very big research paper. I've only taken a small snapshot, but basically they said absolutely yes. They built a polygenic risk score of hundreds of thousands of genes on the microarray, because it was the GWAS studies. There were like 300,000-400,000 SNPs that they looked at. They built a score and they found that the score could predict who was more likely to have diabetes. But if you look where my red circle is in number B, they could also predict pre-diabetes amongst those who did not present with any insulin resistance or any glucose dysfunction. They could predict who was going to be pre-diabetic. And they could also predict, in the one in number A, at what age that individual would likely be diagnosed. What's interesting here is with each standard deviation of the polygenic risk score, it was associated with a 23% increase in the odds of a pre-diabetes diagnosis. So that's pretty significant for something as kind of broad as a GWAS study. All right, let's have a look at another one. This one is coming from the sports space. I've tried to mix it up a bit today, and we're talking about VO2 max. So VO2 max is really, how do we respond to a training program, and where does our kind of potential, our exercise potential live in terms of our response to training? In this GWAS study, Bouchard is one of the preeminent sports genetics researchers. They looked at 324,611 SNPs, and what they found that of that 300,000 and plus, 21 SNPs explained 48.6% of the variance of how we respond, how our VO2 max responds to exercise program in a base population. And you can see from the image, it's very clear. As the SNP score increased, so did the VO2 max training response to an exercise program. So we can see here, again, the GWAS is very useful in terms of being able to look at something like response. But generally what's happened, and this is a lot of the confusion in the industry, is that everyone talks about the polygenic risk score. And when we talk about a polygenic risk score, and I've got a lovely table to share with you, we're usually always talking about using GWAS studies with hundreds of thousands of SNPs, building scores, and looking at a case versus control. But there is another way to do polygenic research, and that is called the total genotype score. Now, when I looked at the literature, one of the issues is this huge confusion in how they name the research, because often what is actually a total genotype score will be referred to as a polygenic risk score. But by the end of today, we are going to have a very clear idea of the difference between polygenic risk score and a total genotype score. So a total genotype score is very different. In a total genotype score, we have a hypothesis and we choose the SNPs. Those are the two big things. So we have a hypothesis. What is the impact of endurance training on VO2 max response would be a hypothesis. And then we would go and look at the literature, and we'll talk about how we do that. And we would choose a number of SNPs that have been shown in the literature to impact VO2 max. And we would build from that, and I'm going to show you some examples, a total genotype score. So what happens, and I'm going to show you a lovely table, I'm getting to my table, is when we build a total genotype score, we're not just looking to see, is there a SNP that shines and pokes its head out of hundreds of thousands of others? We actually want to score it. So we can score it in two different ways. There's two different methodologies that are very well validated, the weighted methodology and the counting methodology. Now, the counting methodology is very easy. I'm actually going to show you an example of both, where essentially we count the alleles. So if you've got a C, a CT, and a TT, and the TT is the impact allele, you'll give CC a 0, CT a 1, and TT a 2, and you put all your SNPs together and you count your alleles and you create a score like that. In a weighted total genotype score, it's different. And it comes to a point which I'm going to raise, which is, should we be counting all SNPs equally? All right. So think about that, because we're going to talk a lot about that when we talk about 3x4. And I'm going to show you an example of two different research studies, both of which used a different methodology. So let's have a look at this one. This is a weighted total genotype score, back again in the world of sports, where they looked at genes associated with muscle injury and injury etiology in professional soccer players. They used 122 professional sports players, and they only looked at 6 SNPs. So there was a hypothesis trying to see what the role of genetics was in muscle injury and the etiology of injuries in soccer players. And they had a very specific list of SNPs that they were interested in, that they already knew about from previous literature. And what they did in terms of weighting is they said they're going to give 2 points to the worst genotype, so the one that would increase injury. They're going to give 0 points to the most protective genotype, and they were going to give 1 point to the one that was a heterozygous, right? And what they found, and you can see this in the image, is that the total genotype score had a cutoff point at 45.83. You can see I've put a red line there to make it easier, so that all the players who had a lower score below that were less likely to be injured. But when it went over that cutoff point, you were more likely to see injured soccer players. So the total genotype score beyond the cutoff had an odds ratio of 1.91, where they would suffer an injury, and 1.19 is a decent odds ratio when compared with the players that had the total genotype score. So in this one, a lower score was actually preferable because of the way they weighted the genotypes. Okay. Here is an example of an allele count. This is to do with diabetes. Very simple study. They looked at, they wanted to know, does a genotype score help to determine the prediction of type 2 diabetes when you add that genotype score alongside everything else that you would normally use to predict diabetes in an individual? They chose 18 SNPs that in the literature had shown. They used the framing and offspring study. They had 2,377 sample size, which is great. And the genotype score was calculated from the number of risk alleles. So 0, 1, 2, exactly as I explained before. And as you can see from the genotype score, that the incidence of diabetes increased as the genotype score increased. So it was a good tool to use to be able to predict what whether an individual is more likely to have diabetes. Okay, that is really the polygenic scores. Now, going back to what I said about the total genotype score, if a gene comes up in the GWAS study, like FTO, and we think, well, this is interesting, something is happening, even if we don't understand the biology, something is happening that we need to know about here. How do we take it forward? And this is what usually happens. It now goes into the phase of candidate gene studies. So a gene now has been chosen as a candidate, or what we call association studies, where we can now go and try understand what role is this gene playing in, what is it doing, and what is it associated with. So we might have a broad idea of diabetes, but is this particular gene really associated with diabetes? So this is just a very crude presentation of what an association study is. Marker 1, you could use the other word, it could be SNP for marker 1, and it's got allele 0 and allele 1. One could be a C, one could be a T. And again, using a disease group and a control group. So we might have a diabetic group in the red and a non-diabetic group in the right. And then we're going to test for this particular SNP and see how the alleles come up. And you can see here, allele 1, allele 0 came up a lot in the diabetic disease group, whereas allele 1, which is the red star, came up. And so immediately we can see that what they call allele 0, which is our yellow square, is associated with the disease. So this is what we'd call an association study. And pretty much most of the studies you have seen will be association studies. Now, just before I go into a couple of examples of association studies, I want to just give you, I've given you 2 tables today that if you ever wanted to go back and remind yourself the difference between a candidate gene or association study and a GWAS study. Candidate gene is a technique that investigates an association between a SNP with the pre-specified genes of interest and a phenotype or a disease state, whereas the GWAS investigates an association with genetic variants across the whole entire genome with the particular disease. So testing, it's analyzed, the variation is analyzed within a specific gene, so we know exactly what we want to test, whereas in GWAS, we're testing a whole broad number of genes. In candidate gene studies, the statistical power is much more powerful. We can believe in it a lot more. In GWAS, it's a very low statistical power. Remember my idea of fishing expedition. It's good to find them, but we shouldn't make any recommendations on them. Do we need any prior knowledge for candidate gene studies? Absolutely. We want to know what other research has been done on it. GWAS, not at all. And selection of genes is definitely needed for the candidate gene studies, not for GWAS. Okay, let's move on. Here's an example of a candidate gene study or an association study that I have taught on quite a lot. It's ApoA2. Again, a very interesting gene because it's been quite hard for us to understand exactly what it does. We know it's an apolipoprotein gene, but what's interesting about it, some of the best research has been done in terms of association studies on ApoA2, and it was done by 2 of the most brilliant researchers in this field, Jose Ordovez from Tufts University and Dolores Carrera. And why this is such beautiful research, and in my old course, I teach this as a perfect example of perfect research, is because the sample sizes that they looked at are huge. So there's like 2,000 to 3,000 in every study. The other thing about it is they replicated the study, in 5 different ethnic populations. So this isn't a case of just looking at the framing, and they did it in Europe, they did it in South America, they did it in 5 different ethnic populations, and they got exactly the same results. So that, on so many levels, is a fantastic research. Now let's look what they did. They had a look at 3 variables: the ApoA2 SNP, saturated fat, and BMI index. So, we could say, well, saturated fat is complicated and they should have looked at fatty acids, and BMI is not a great measure. But remembering they were looking at 2,000 to 3,000 in each sample size. So this is epidemiological in a large degree, right? And what found was essentially, and the reason I love the study is if you look at number C, when only looked at the gene against BMI, they found that there was really no difference for the CC, the TC, and the TT in terms of a different BMI in the Framingham study. This is just the Framingham we're looking at here. When we added in nutrition as a 3rd variable, suddenly the results broke open, and you can see that indeed, that when we started looking at saturated fat, they found that actually how much saturated fat you were having in your diet would determine your likelihood of having a high BMI based on your genotype. And this is why when we look at nutrition research that doesn't have genetics, we know we're missing a piece of the puzzle, and vice versa. When we're looking at genetic research that doesn't have nutrition in, we're missing in the piece of the puzzle. Anyway, don't want to carry on too much here, but this is a perfect example of an association study around a candidate gene, where we now know the gene's important. Now we're trying to get into the deep dive of, well, actually, what is this gene variant doing? Okay, love the slide. Want to thank my outstanding designers, Vincent and Robin. They just make the most beautiful slides. And I asked her for a slide that showed celebration. That's all I said. And she gave me this. And the reason I did this is I want to show you what my most favorite kind of research is. So I've spoken about GWAS, total genotype score, association, candidate gene. But at 3x4, there's another kind of study that we absolutely love. We get very excited in the science team. And that is when we can start talking about biochemistry, physiology, all right? And that means that we can tell what the biochemical impact is that the SNP is having. Because just knowing ApoE is associated with BMI at a particular saturated fat intake is not enough information for us at 3x4 Genetics. We believe that everything begins and ends with nutritional biochemistry. And because of that belief, we want to know what is the impact of a SNP on our physiology, biochemistry, enzyme, neurotransmitters, hormones. Is it changing anything in any way? These are the hardest studies to find, but this is where we always focus on. So we are looking at everything I've showed you up until now. But we really, really, really want to know that if you have the MTHFR variant and you have a TT genotype, that your enzyme activity has been decreased by 50 to 60%. So here's an example. One of my favorite genes, you know that it's one of my favorite genes, quinone reductase, NQ01. Phase 2 detoxification. This is a reductase, a cytoplasmic reductase enzyme. It reduces reactive quinones to less reactive and also less toxic hydroquinones. And if you just can imagine that beautiful picture of phase 2 detoxification, and what it's able to do is it's able to recycle these quinones when our phase 2 detox is not working optimally and it's heading towards that DNA adduct. Quinone reductase picks it up, reduces it, brings it back to the beginning of phase 2 detox and gives it another chance to be able to be detoxified and excreted from the body. So we love quinone reductase. But there is a SNP that is a CT SNP, T being the impact allele. And the other thing about quinone reductase, it activates NRF2, the transcription factor. So we love quinone reductase. Okay. Unfortunately, what we found in the research, remember my favorite study, is that when you have a TT genotype, the enzyme has little or no activity. So the TT genotype has 2 to 4% activity of the CC, which is the most common genotype. That's pretty much hardly anything. And the CT genotype, your heterozygous, has a 3-fold decrease in enzymatic activity compared with the CC. So this is the kind of study that we celebrate, because it's giving us such useful information when we're sitting with a patient and we're trying to make our clinical decisions about how to manage. So it's one thing to manage detox. It's another thing to know that quinine reductase, if you've got the TT genotype, is not functioning almost at all. So how else can we switch on phase 2 detox and support it? And how else can we really upregulate NRF2, our transcription factor? So these are the kind of studies that we are always looking at, because if we know what's happening at a biochemical level, it's really informative to us. Okay, so that really gives you the background. Oh, didn't bring water with me. That's what gives you the background on all the studies that the science team at 3x4 will be studying to be able to get to the point that I am now, which is, what is the 3x4 methodology? And remember, I started my talk with a slide about evolution. And 6 years ago, when I started my journey with 3x4, and I was thinking about, how do I build a better test? How do I deliver a better science? This is the point that I was at, which is, I know all the stuff, I've been reading this literature, but how do we do things better? So it wasn't enough that we just built a total genotype score with an allele count. I really thought that there was a better, more evolved way of doing things, and I'm going to share that with you now. So as you all know, we base our methodology on the concept of a pathway-based approach. And the reason I wanted to make this clear before we go in, we are not a polygenic risk score in any way, and you'll see why. I don't even want to call us a total genotype score, although that is the closest that we are, for sure. And so we call it a pathway-based approach, because what we're interested is in biochemical pathways. Now, remember, every single study I showed you up until now was disease associated, right? Diabetes, obesity, arthritis, hypertension. And we are, obviously, our background is in systems biology, integrative medicine, and functional medicine. So for me, there was no value in building a total genotype score associated with a disease endpoint. That would be going backwards for me. So I wanted to go back to the absolute core, which is biochemistry, and look at what were those biochemical, or sometimes you'd metabolic processes that we could take was a really excellent methodology, which is kind of total genotype score, not polygenic risk score, and apply it to biochemical pathways. And that is why at 3x4, we talk about pathway-based approach. So there's one unique thing we do already, which is we focus at the biochemical level and not at the disease level. And you can see that from the 36 pathways that we study. This is really another, as I said, another table, which is really useful to go back to, where I compare the polygenic risk score, which people often think is what we use at 3x4, with the 3x4 pathway-based analysis. What is the core differences? How we select our genes. We don't use GWAS, we use Darwin. I'm going to talk about Darwin. We don't measure risk. Very important. We do not measure risk. We are not a risk company. We never talk about risk. We're not about medical genetics. We talk about impact. Calculations, we do not use odds ratios. We use Darwin, and I'm going to tell you how we do it: scientific validity, clinical utility, machine learning, and thresholding, which I'll introduce to you. We do not look at disease endpoints. We look at optimal biochemical and metabolic processes. We do not use 3x4 for disease risk calculations, not at all. We do use it to try get focused clinical intervention. And what kind of insights do we get? Polygenic risk scores get scores that are really good for screening, case control, and predictability. But at 3x4, what we're trying to understand is how are genes impacting these biochemical and metabolic processes? And as a result, what kind of insights can we get for diet and lifestyle recommendations? Okay, So now that we've got kind of our pathway-based approach, how do we choose the SNP? And I always say, if you're ever reviewing another genetic testing company to use them, the first thing you want to ask them is, how do you choose your SNPs? Because not every SNP is equal. We know this. ApoE and COMT and MPHFR are going to be, and quinone reductase are going to be different from the other 150 SNPs that we're testing. So for me, counting alleles just never made sense, because how can I count an ApoE34 allele in the same way that I count another allele? So I needed a better methodology. And that is when we get to Darwin. And of course, we call Darwin Darwin because the concept is only the fittest genes will survive and land up in the 3x4 blueprint. So let's understand, Darwin was not my invention from scratch. It didn't come out of nowhere. It's based on the ACCE model, which is a very highly validated model that is used in clinical research across the world. It looks at 4 different things: analytical validity, accuracy of the genotyping, scientific validity, how good is the science, clinical utility, can we use this in clinical practice, and lastly, ethical, legal, and social implications. So for Darwin, I'm going to focus on 2 and 3, but of course, the company as a whole is very focused on making sure we've got the most accurate genotyping, as well as how we manage our ethical, legal, and social. So these are the 2 that are key concern to me: scientific validity, how good is the science, clinical utility, how clinically useful is it? And I often say there's many genetic testing tests in the marketplace, and pretty much all of them will focus on scientific validity. What you need to understand, and which is much harder, is clinical utility. Right. We have this brilliant team, both the science, the clinical, and our design team, who have built you a tool. And I am actually going to show you a couple of slides of the tool, but the tool will be available for you on the community when we post this presentation, which at any time you'll be able to review or share or go back to, which essentially gives you the steps we take in deciding how we incorporate a gene. So the first thing is scoring of SNPs. So before we started Darwin, I had spent like a year pulling together a list of gene variants that I thought were interesting for 3x4, and I think there were 1,400 in my list. But just because I have, and they were like scientific validity in many ways, just because I have 1,400 doesn't mean we're going to have a 1,400 in our genetic test. So how do we test those 1,400, which came out of association and gene candidate studies, to see which genes were the right ones for 3x4? Step 1, all right. Darwin basically has 8 criteria. 4 criteria for scientific validity and 4 criteria for clinical utility. In each of these criteria, a SNP will be scored by one of our science team, and we have 4 geneticists in the company, plus me, so that's not including me, because so that's how many geneticists we have working on Darwin, and it was all done manually by hand, right? The first thing we do is we evaluate the quality and type of the literature. How many studies? Was it replicated? Sample size, in vitro, in vitro, everything that you know about good science. Then we look. So I'm not giving you all 8 criteria. I'm kind of just giving you a more broad view of how we do it. Then we look for those magical celebratory studies. Can we see the functional impact of the SNP? Can we find research that tells us, if I have the SNP and I have the TT, I have 2 to 4% of quinone reductase activity? Then what we want to do is understand the SNP's role in the relevant pathway. And the word relevant is very important here, because we know that gene variants will play a role in multiple pathways. So if we think of COMT is going to be in estrogen, it's going to be in hormone, it's going to be in mood, it's going to be in methylation. So if we think about ApoE, it's going to be in inflammation, it's going to be in mood, it's going to be in cognitive function. So, sorry, not mood, in cognitive function. So when we look at a SNP, we don't just score it and say, is there literature? We say, is there literature in that particular pathway? So before we started Darwin, we determined our 36 pathways that we wanted to investigate. And then when we have a SNP, we will look at it in reference. So some SNPs we will score in 5 or 6 pathways, some in 1 pathway, some in 2 pathways. But whatever it is, the literature has to be in that pathway to get a score. It can't just be, this ApoE is associated with cardiovascular disease, and therefore we take ApoE and put it in the inflammation pathway. We can't do that. So we're very strict about the SNP staying in its pathway lane. The next thing we want to understand is there some kind of clinical impact, some kind of phenotype that the SNP is going to impact, whether it's lipid metabolism or glucose homeostasis or detoxification or inflammation or hormone levels? Is there a phenotype associated with this thing? Because if there isn't a phenotype, it's not going to have clinical utility. And then the next one would be, is there some kind of intervention that as a clinician I can do? Because again, if it's interesting and it's valid, if I can't do anything as a clinician, why would we test this? Second lastly, are there biomarkers or measurables that are associated? So it might be a direct measure, like IL-6 being able to measure IL-6 protein, or TNF-alpha being able to measure TNF-alpha. So sometimes it's direct measure, but sometimes it's an indirect measure. And you'll see that in our clinical guide, where we will give you what are those indirect measures associated with the SNP that you may want to test in functional or lab tests. And lastly, my most favorite one, will this genetic variant influence your treatment decisions? Will it change your mind or change your focus on how you will treat your patient? Because then we know that it's really clinically useful. And so we score 8 criteria, and we come up with a SNP score. And if it gets over 50% for the SNP score, it goes into our genetic test. If it's under 50%, it doesn't. And I'm sure you've been wondering, maybe in the chat, you could put there, I know you're familiar with the pop stars in the genetic report. What percentage do you think a SNP needs to get and score in Darwin to be able to get a pop star? So all of you can pop it into the chat, and we'll have a look at it later and figure out. I know some of you might know the answer if you've been paying attention, but let's see how many of you can have an answer of what percentage do you score to get a pop star in the report. And now you understand why certain SNPs will get a pop star and others won't, and also why some SNPs will be in or not. Then what we do is we hand it over to our brilliant biomedical engineering team, who have built a machine learning model where they basically take the score and the cumulative impact of all the SNPs in a pathway, and they simulate all the possible SNP combinations for a population. And we use a database that is available in open source. And from that, we're able to convert the score into an impact threshold. And you can see the image there with the colors. You're now starting to see purples and blues and yellows. And from that impact threshold, we can now determine what your individual impact will be based on that thresholding. So where in that scale do you come out and you get your beautiful report, your patients get your beautiful report, and it'll have the purple, the blue, and the yellow. So I hope that gives you an idea how we go from the total genotype score through to the report. And of course, we have outcomes. And when we get our report, which now has those colors that have come up, the very high, medium, and low, just want to make sure I've got a few more minutes left, we can now talk about precision functional testing, personalized diet and lifestyle recommendations, and what we talk about targeted supplementation. Okay. This slide I always put up because it always comes down to that last point in Darwin, which is, can we use genetics to make more personalized interventions that this dietary guidelines cannot do? Okay. I'm running out of time and I want to make sure I keep my time, but I did have a little giggle when I was going back and looking at my report. I want to talk about 2 SNPs that we can look at scientific validity and clinical utility. GSTM1 is your phase 2 glutathione transferase. And we know that when we have a gene deletion, it knocks out the gene completely. We have 0 activity of the gene. So we know from the science that there is an impact, there's a biochemical impact, and there's lots of research. And we know that for clinical utility, knowing GST is going to be very useful. And the reason I've put in this slide, so I sent this slide to the designers and they came back going, please let me redo the slide. And I was like, leave the slide. And the reason I wanted to leave the slide is because it came out of the Manuka Science course from 2015, the first nutrigenomic course I ever built with Christine Horton, and I made all my own slides. And this is one of my slides. So I wanted to show you how lucky you are that we have progressed in our slide making, because this is what all my slides look like. Well, the content was great. The execution of my slides was pretty awful. But anyway, the point being that knowing GST can be super, super helpful. Here's a gene that comes up often, the LCT gene, the lactase gene. And it caused a lot of controversy because LCT did not pass Darwin, is the bottom line. And I'm going to tell you why. So when we scored LCT, which is the lactase gene, it's actually called by a different name. It's called MCM6. The lactase gene, the LCT gene, is actually very difficult to test on the array. So we test the MCM6 gene, which is in what we call linkage disequilibrium. In other words, they're inherited exactly the same. So by testing this one, we can determine what the LCT gene is. The LCT gene number is actually 13910CT, and the MC, and it's upstream from lactase. So by testing one, we get the answer for the other one. And it gives us information about the enzyme lactase, whether as an individual we are expressing that, that protein and making the enzyme lactase to be able to break down lactose. And we know from the literature, pretty much every single study will tell you that if you have the CC genotype, it may predict the presence of lactose intolerance, remembering that the ancestral version of the SNP is not to have lactase. We were not ancestrally born with lactase. We could not tolerate lactose. And then when we started settling down in Northern Europe and farming and having animals that we needed to survive off their milk, we developed this enzyme that could break down lactase, right? So CC is the original, the ancestral, and that's why it comes first, and TT is the new one. Now, here's why it failed Darwin. So it failed Darwin because it passed scientific validity, because there is so much research on this gene, and it has been probably for like 15, 20 years, but it failed clinical utility. And that's why it didn't pass. And here's the reason why it failed clinical utility. And I'm just going to show you one research paper to demonstrate. Remember what I said before, the CC genotype may, and I put here may, predict the presence of lactose intolerance. And this may didn't come from me, it came from the research paper. Have a look here. On the left, I've got lactase activity. So that is the lactase enzyme. We've got on the lowest one is the CC, then TC, and then TT. And the CC is the one that is associated with lactose intolerance. Now, where this dotted line across is the threshold where we believe we don't have enough lactase enzyme and therefore we are lactose intolerant. So what they're basically saying here is everyone under the line should be lactose intolerant and everyone above the line should be lactose tolerant. Now, what happened is many years ago in 2014, Anneli Smith, who's head of clinical mentorship, and myself and two other dietitians had a clinic in South Africa called the Center for Translational Nutrigenomics, where we were testing every single patient that came to our clinic. And we were testing this the LCT gene. And we soon realized that the information that we were getting from the report was not manifesting phenotypically. So someone would come in and we would be very clever and say, well, we've looked at your report and you should stay away from dairy because you're lactose intolerant. And they would look at us and go, but I've been like had double latte before I came over this morning. I've been living on dairy. And Anneli and I started getting very, very perturbed about why we were seeing such discontinuity between what the genes were telling us and what the person was telling us. So when we went back to the literature, when we built Darwin, this is where we started seeing, first of all, you can see the high variance in the results. You can see how far the spread is. But look at the open circles. I hope you can see on the screen. Open circles indicate individuals who self-reported being lactose intolerant. So these are the ones who joined this study and said, I can't tolerate milk. All right. Now, what happens if you have a look again is, yes, there are 3 open circles in the CC genotype of people that were lactose intolerant, but there are about 10 of them that are supposed to be lactose intolerant, but they're not lactose intolerant. And in fact, if you look at the TC, there are 3 individuals who are lactose intolerant, but actually they were above the threshold of lactose intolerance. I'm not going to spend any more time here, but if you study this slide, you will see that it is absolutely impossible to predict lactose intolerance using the LCT gene. So yes, scientific validity, failed clinical utility, failed Darwin, not in the 3x4 genetic test. Okay, my last slide before I get to, please don't leave. I want to introduce you to the team. Right, the bottom line, clinical translation uses both SNPs and pathways. We never only look at the pathway page when we are reviewing a report. I look at the pathway page to see where the purple is, see where it's impacting, go upstream. Then I go look at the gene page, the SNP page, and then I go into the clinical guide to understand what is the role of the SNP in that impact. This is not binary. This is not, I only look at pathways and I only look at SNPs. This is being the best nutrigenomics practitioner, uses pathways to guide us where to focus our attention, where to start. And we use SNPs to be able to understand the fine-tuning of the personalization. Second, grouping genes together increases the power for us to have greater insight into our patients. And third, no matter what we do with genetics, we always have to remember that clinical translation is paramount, absolutely paramount. Okay, I believe that is, before I forget, in addition to this magnificent tool that my team built for you that describes Darwin pathways, how we do it, thresholding, my team has also built a document for you called our top 100 references. So we often get asked the question, which reference, can you send me some references for 3x4 so I can see how you do things? And it's always difficult for me to say, well, I don't know, like we've got thousands. So I gave a challenge to the team, in particular to one of our geneticists, Helene, to come up with our 100 top references, which we've put into a document for you. It's available on the community. It will be available on community. And we've broken it up into monogenic and polygenic, precision nutrition. You will see it's a really magnificent document, and a big thank you to my team for doing an amazing job on this. So I am going to stop sharing, and I am going to ask my team to unmute and make sure we can see your faces, because you need to understand the amount of work that has gone in to everything that I've shown you today is extraordinary. I hope if we only learn one thing today, it's the appreciation. I remember when Helene, who's unfortunately in Italy at the moment, so is not joining us today, when Helene joined the company for 2 years, all she did was work on Darwin. She literally spent every day reading literature. It's how we kind of put everyone through their, to get the kind of 3x4 t-shirt is literally Darwin, reading literature, scoring, reading literature, scoring. So unfortunately, Helene's not here today, but I am going to introduce you to my magnificent team. I'm not going to introduce them. I'm going to ask them to introduce themselves, what they do at 3x4, what their background is, and why they love working for 3x4. So I'm going to start with our head of product, Dr. Gerida S., if you can take the stage.
Thank you, Yael. I almost said good evening, but I realized it is not evening on your side. So like Yael said, my name is Dr. Gerida S., and I am the head of product. My background is in medical genetics, specifically cardiac genetics. So I did my PhD in looking at the genetics of sudden cardiac death syndrome in athletes. And what we found way back when was that with genes that were causing the disease, there was a polygenic background that played a role in the manifestation of the clinical phenotype of that disease. So when I joined 3x4, I had that knowledge, and I was very excited to put it all into practice. I also did my postdoctoral fellowship at Harvard Medical School in Boston, Massachusetts, where I focused on cardiac regeneration. And ever since then, I've been involved in product development, scientific product development mostly. So what I do at 3x4, as the head of product, I am responsible for overseeing everything product, that is from the conception of a product to the consumption of a product. But where I am mostly involved is the scientific research and development that contributes to a product. And for scientific research and development, there are two things. The one is technical science. So that involves everything in the laboratory, as well as our arrays on which we do our genetic tests. So as you all know, we've got a custom genotyping array, which we've built. I head up that effort. And then the second R&D component is then the genetic science. So I have contributed on the intellectual property of both our blueprint product as well as our new GeneFit product. I think that's it in a nutshell. Why I love working.
Yes, why do you love working at 3x4?
Why I love working at 3x4. I think we're in this age of social media where everyone is an expert that has a good social media presence, but a lot of people is so full of talk. And I don't like talking very much. I like doing. And what I like is actually taking the science, taking all the knowledge that we gain and building that into products where we can see it actually makes a difference. So taking science and bringing it to life. That is why I love working here.
And I just want to add that Gerida and Mariette, who you'll meet now, have headed up our GeneFit, our sports genetics initiative and project, and is also a world-class triathlete. So she's never going to tell you that herself. Thank you very much, Gerida. Annelie, I think everyone knows you, so keep it very short and sweet. I think everyone knows you, but please say hi. Oh, come back to the screen, Annelie.
I apologize. My internet decided to let me down. I hope you can hear me. Okay, so I know a lot of you know me. So I head up the wonderful clinical and mentorship team, and I am a certified functional trained dietitian. And I have been, in my previous life, I was in private practice and worked in many different places. I worked in private practice for, I think it was almost 30 years. But from 2002, I started working in nutrigenomics. So that's how Yael and I know each other. We used to bump into each other at places where everybody thought we were a little bit crazy looking at nutrigenomics, but we were working in this same sort of space and kept bumping into each other. And I've been involved with 3x4 since before the beginning, but definitely since we launched in 2018. And my role really was on the clinical side with the development of some of the product, obviously the recommendations, the clinical guide, the resources that you see. I've interpreted thousands of reports, so a lot of us. So if I speak to some of you and you think it, it's like learning a different language. So I've just done my 10,000 hours in interpreting it, and of course, also then mentored many practitioners in the process. So
why do you love working at 3x4? Most important question.
So why I love it is I live my best life. I do what I love. I can sit all day, every day and look at genetics and help practitioners solve puzzles and really help them improve their patients' lives. And because I'm on the forefront and I see a lot of this, I really see a lot of success stories. And of course, 3x4 is such an innovative and forward-thinking company, and I love working here. So that's my story. Thanks.
Thanks, Annelie. Right, Mariette, if you can step forward. Mariette Kuhn.
Hi, everyone. I'm the head of engineering at 3x4. I've got a master's in biomedical engineering and 20 years of experience in research and product development, specifically in the field of biosignals. I did a lot of work in prosthetics as well, not only genetics and biosignals, but prosthetics as well, which is a fascinating field. I'm responsible for all the algorithm development, the technical architecture development of the product, as well as the machine learning and data science of all the data involved. with our products. Also the co-creator of GeneFit, as Yael mentioned, also an avid triathlete, and that's where it all began. I love working at 3x4 because we want to be the best in the field of genetics. And in order to do that, I need to be on my toes, so that we always have the most advanced technology. And that also gives me the freedom to do whatever is needed to transform the newest research that the science team is doing into a user-friendly product. Yeah, and that's always cutting edge and a lot of fun. I feel like I'm playing, I'm not working.
Thanks, Mariette. And again, world-class triathletes. The both of them are very humble when it comes to how brilliant they are in triathlon. Right, Nikki, I know you're very well known by everyone, so let's quickly introduce you. I'm sure everyone's had some input from you.
Yes, hi everybody. I'm Nikki Grant, for those of you who don't know me and haven't met me before. I am lead mentor and nutrigenomics clinician at 3x4 Genetics. I'm a registered dietitian with a BSc in nutrition. I am a functional medicine certified practitioner with almost 20 years of experience in nutrigenomics in practice, and having used many different tests in my practice, I became very frustrated, like many of you, about how cumbersome they were to translate and how time consuming they were. And then 3x4 was born, and this was the solution. That pathway-based summary page that Yael mentioned, it's page 8 for anybody who doesn't know, in the PDF, became my go-to for the snapshot insight into where to start with a patient's treatment and intervention. And it made genomics, nutrigenomics, come to life for me. Why do I love 3x4 and love working for 3x4? Because I love to make science simple and understandable. And I may or may not be known as the analogy queen. And yeah, so that's me.
Thank you, Nikki. Right, Clara, please step up.
Hello everyone, I'm Clara van Skalkwijk. I'm a researcher in the product team, and I'm also responsible for our data quality control. I did my master's in biochemistry with a specific focus on systems biology. And I love working at 3x4. I think my answer is sort of the same as everyone. It's because we translate scientific knowledge and we make it accessible and actionable. And we are continuously learning and improving our product.
Thanks, Clara. And I have to say, you know, Clara, Chantel, who you'll meet now, and Helene, who's not yet, came almost straight from university and have just grown to be so extraordinary and have almost like running their whole sections themselves now. So quite extraordinary. Right. I would like to now introduce Chantel.
Hi, everyone. I'm Chantel Rousseau. I'm also a product researcher. So the main focus of my research is sports genetics and human performance. So a lot of my research has been going into our GeneFit product. So my background is I have a bachelor's in biochemistry, also specifically focusing on genetics. And yeah, the reason why I love working at 3x4 is definitely the team that I get to work with. It's so inspiring to work with so many brilliant and curious individuals. And every day I get challenged to improve my understanding and also to grow as a scientist. So yeah, that's me.
Very much, Chantel, again. And then before I go to the last person, one of the kind of genetics team members is missing, Helene Herholt, who was actually the first one who joined us. And she's responsible for the tool and also for the references, working with everyone else, of course. And she's also, I think she's got a master's in genetics. And she's also an IFN Academy qualified functional nutritionist now as well. So really just want to note her extraordinary contribution. And last but not least, I'd like to introduce you to Di. I think you're on mute, Di. Yeah.
Hi, everybody. My name is Diane White. My role at 3x4 is in the capacity of sports scientist. I come from an endurance sports coaching background, having worked with a variety of athletes from novice to world-class level, as well as 2 professional cycling teams. My role at 3x4, I advise and provide support in translating the genetic science into actionable recommendations in the activity pathway. which includes training, response, recovery, and injury, as well as creating content for our new GeneFit athlete optimization platform that is currently in development. And what really excites me about working at 3x4 is how genetic insight can drive athletic performance through very personalized workout programming, as well as nutritional support. And it's something that is taking not only sports performance, but also just healthy activity to a new level. And that's quite something in the world we live in nowadays. So that's me. Thanks.
Thanks, Di. And like many on the team, they come to us for working just a few hours, and then they never leave. And we don't let them go. So Di is one of the more recent that joined us, and I don't think she'll be leaving anytime soon. I want to thank everyone. And of course, it's not just the brilliant science, product, and clinical team. It's the design team, the web team, the tech team, the ops team who make everything happen at 3x4. But I thought it was really important that you know that it's, I get to do the talking, but it really is this extraordinary team who is working the long hours behind my back. So thank you everyone for joining us. And I hope you enjoyed the tool. And if you have any questions, please post them on community or send them to us and we'll post them on community. Thank you all for staying till the end.